things. It makes installation fairly straightforward. Our installation is fine. Conda is sometimes buggy, so if anybody's dealt with Conda. And there is a Read the Docs, a documentation site that's linked from there, so we have quite a bit. We'll actually show that in a few minutes. So I'm just going to, I slightly modified this slide from Christine, just to say, you know, we're using K-mers, we're using reduced amino acid reduction, and that gets has some advantages in terms of speed, so you can match these K-mers really quickly, at least in theory. And we can put this into machine learning, which is great. We've got we've already used machine learning and to do this kind of training that I'll show you, but there's opportunities for data integration here. And so one of the places where we're really interested in going is taking some of the other kind of multi-omics data, for instance, from EMSL projects or other projects, and feeding that in along with the sequence analysis and building machine learning models that will be better at functional prediction. So I've actually funded under DOE a university proposal to do some of that, and hopefully getting to that this year. And then really what we're going to be talking about here is capturing those distantly related sequences. And I've got a really great example of that, where this is kind of I would say a motivating example of where this kind of K-mer-based approach with amino acid reduction has really worked well. So some of the gaps in this protein annotation space are, you know, annotations are based on a limited number of methods of similarity determination. Many genes and proteins, there's the dark matter, right? Have no useful annotations because they're not similar enough to anything to kind of fall next to something that already has a functional annotation. Some functional families are divergent in sequence space. I've made this big because this is kind of one of the motivating factors for the original. My head is really big. For the original, direction or the original experiments that was done here. And then we have a couple of examples from other places that are a little bit different. So transporters are a place where you can pretty easily identify that it's a transporter. It's much more difficult to identify what that is going to be transporting, the substrate that's going to, or substrates to be transporting. So we have kind of plans to go in that direction. The motivating kind of example here is something that I did a number of years ago now. This is to capture ubiquitin ligase mimics. So ubiquitin ligases are very common in eukaryotic systems. They perform a number of different functions that kind of modify protein. So they are modifiers of protein function. So the ubiquitin ligase, usually fairly specific, it will go in and alter one specific set of proteins by adding ubiquitins or be part of the process that adds ubiquitins and change things like signaling pathways or metabolism or things like that. It's a completely normal process, and pathogens, especially bacteria and viruses, have developed ways of interfering with those natural systems. And so there's a large collection of ubiquitin ligase mimics, and these are either convergent or drastically divergent evolution, where these bacterial proteins and viral proteins have similar functions. They all do the same ubiquitin ligation. And they have very divergent sequences. So that's captured in from this paper from a number of years ago now. So you can see the sequence alignment down below is just like, you would not be able to look at that and say that these are similar at all, right? But the structures from each of them in these important regions are very, very similar. And so this is a motivating example, because it's a place where you don't have a blast or an HMM that can capture all these. Actually, there's multiple HMMs that capture parts of this large functional family. And so what we were interested in doing is developing a machine learning method to predict ubiquitin ligase effectors, and I developed this method using, I think I called it reduced amino acid fingerprinting, amino acid reduction. And it worked really well, and we're able to get a nice model out that predicted ubiquitin ligase like effectors, and it was written in piecemeal, and so we had bits and pieces sitting all over the place. You could kind of put it together in a supplement, and it, you could run it through all the way from start to finish, but it took a lot of work to do, and it was very difficult to tailor it to your own application. So Snekmer was developed to make that process very painless. And so the idea is that you can gather up your own set, as Christine was talking about, you can gather up your own set of related sequences. This could be from a sequence search. You might go to NCBI or Uniprot and say, hey, I want sequences that are from this functional category, or sequences that are in this family or something. Or it could be from, experimental investigation. One of the places we've used it is in activity-based protein profiling. And so that's where you have a modified substrate, or you have a chemical substrate probe that you bind to an enzyme, or you bind to a sample, and and the enzyme pocket grabs it, and then you cross-link it, and you pull out everything that binds to it. And so you're saying, everything that binds to this substrate, I want to know about what it is. And we've used it, applied it to those sets of proteins, which are very experimental based, and so to try to develop models to tell you what's similar with these things. So I'm going to just go into the demo here, and I will. Oh, so is my head back up there? I got to share something. So I'm going to share a terminal. Okay, just a minute. And show you kind of the process that you go through once you have that set of protein sequences. So this is from that ubiquitin ligase example. The way that I got those is I went into several different databases that capture these kinds of viral effector or pathogen effectors, and I grabbed out all of the ones that had been validated as ubiquitin ligases, and I collected those into a set of sequences. And then I also took, as kind of a counter example of the training set, the negative examples. So I took another set of known pathogen effectors. So effectors is just a protein that goes in and does something to help the pathogen, that weren't ubiquitin ligase. So this is actually kind of a tricky discrimination, because they're all effectors. Just one class is ubiquitin ligases, and then you have everything else. Sorry. Sure. Okay, so you should be seeing, this as a blank page. So what I have is this how big is that? That's pretty small, isn't it? Let's try to. Okay, that's better. So what I have is a directory where I have two things in it. One is an input directory, which is required by Snekmer. I've already done the installation of Snekmer, and the activation of the Conda environment. That's all part of the documentation that Christine has on the Git repo, or linked in the Git repo. The input directory has our two separate, and it has several other things too. It has two FASTA files. A FASTA file is just a way to store protein sequences, very simple. So I have effector, which is just generic effectors, and I have UB ligase, which is all of the ubiquitin ligase sequences that I collected, and there's 100 and some in there. And the config.yaml, YAML is I think it stands for yet another markup language, which it is. I can definitely attest to that. It is another yet markup language. So we have an example YAML, config YAML as part of the repo, and it basically looks like this. This doesn't probably mean very much, to anyone, but we've specified the length K here. We've specified the alphabet, and there's a decoder. We can actually put the name of the alphabet in here as well as part of the documentation to tell you what that 0 means. In this case, it's the simplest one. It's the hydrophobic and hydrophilic group. So it only has, and it's an alphabet the size of 2, right? You just have either hydrophobic or hydrophilic. And then you have a bunch of other parameters. A lot of these don't need to be changed. They're basically default parameters. But I'll just point out that we have cluster parameters here. And again, this is kind of the default parameters that we've set up to do clustering, which is like unsupervised. And I'll show you an example of that. And then we have modeling parameters. Not really anything in the modeling parameters. We've got cross-validation of 5, and you can set a random state if you want to be able to get the same exact cross-validation if you're like want to be able to replicate something exactly. And so that config.yaml will just tell Snekmer what to do. And so the 3 modes that Christina talked about before, one of them is model. So you just type in Snekmer model, and I'm going to let it go, and it'll go for about 30 seconds to a minute, and it'll spit out a whole bunch of snake-make stuff. But we can talk about what it's doing. Yeah.
I have a quick question about the input.
Sure.
On the FASTA file, do you have a minimum requirement of how many sequences you need to feed?
More than 1.
But you do have a statistics?
Yeah, that's a great question.
Because, for example, in AlphaFold, when they did multiple sequence environment, they found that if they're working with less than 30 sequences, the likelihood of getting correct structure is low.
Mm-hmm.
If they do at the multiple sequence alignments above 100, the gains are very minimal.
Yeah.
So they have
what was the gain that you get from adding more sequences? Yeah, it's actually
so they have like
for
a
our next
plot.
paper.
So they have a plot where they did statistics, how much is input needed to get confident files that they consider.
The answer is it depends. I know that because each different family and each different collection that you have will have slightly different. So you can say, I can put in 100 sequences. If they're all identical, it's not going to be a great model because they're all the same. But if they're all divergent by 10% from each other or from a center, then
yeah, I don't know that much depth. How
yeah, no, it's
an interesting question, partly because of the recoding concept, right?
Yeah, because how much information they need to create a good model, training models that you can apply on your data, right? So
and it would be
how
nice.
much is enough? How much?
Right.
And frequently, so it's to some degree the same rules that apply for any training set you're going to do for the model apply, right? So the more diversity you get in there, that's true positives, the better. But because we're doing the recoding, which can take into account unobserved sequence diversity, right? It may wind up. I don't know. We haven't really tested this out as much as we would like to yet, but it's possible that you can get away with a smaller training set because of that particular aspect of it.
And I mean, for AlphaFold, it might be much more demanding because reconstructing the entire acid while you're working with blocks of amino acids, so maybe UTMAT is much less. But I was just.
Yeah, no, it's a really good suggestion actually. We should have something like that, and
it probably depends on the alphabet you use as well. Like a less complex alphabet may be less important that you have high sequence diversity in it.
So for now, just more than 1.
So for now, what's the minimum? I don't know. I don't know that we've come up with a minimum. Exactly. I would say, as many as you can, but we've worked with things that are like 8 or 10 sequences, and then all the way up to hundreds of sequences.
We do have a couple of the nitrogen families, but I think only have like 3 to 5.
Well,
those ones actually don't perform all that well.
The Taliesin examples that I've done only have about 8 or 9 examples, and they do pretty well at picking up, other things. Yeah.
How does it scale up, like in terms of time of computation, say 50 to 200 sequences?
50 to 200 is, let's see, it depends on what you're doing. So if you're modeling, those are actually fairly comparable because the main time there is actually the K-merization, just translating things into K-mers, and it's not, we haven't optimized that step. So it's all in Python. We've done some things to make it so that it's not super slow because there's ways you can make it super slow in Python. I've found a number of those ways. And also super memory intensive, but we haven't really optimized. So I think if you're talking about training models, 50 to 100 or 10 to 100, there's probably not a lot of difference. If you're talking about clustering, and part of the problem with clustering is that if you're trying to cluster 10 million sequences, most of the clustering algorithms that come in like sklearn they have to see the entire matrix all at once. And so if you have a matrix of 10 million by 10 million, you're basically not going to be able to do it on most laptops. There are ways to parallelize that, but
there's also incremental clustering.
Clustering, yeah.
Yeah. So
yeah, we've thought about looking at.
Actually, so do you look for? So basically, are you looking for basically people to do some stuff for you? Because for example, I asked Christine. I said, hey, you can just very simply automate to find, let's say, K. And the user shouldn't be bothered with that. So I was just, I don't know.
Yeah. Well, we could definitely.
It's a public repository.
Yeah.
Anyone can join in the fun.
So I'm going to just show these results really quick. This is from training, and I'm navigating down these down into this directory structure. But one of the things that we're doing actually not as we speak, but currently we're working on a high order report structure, a report file that will just be there. So you'll be able to go to the report file and find everything that you need to know. You don't have to dig down in this. And so what I'm going to do is open this, and I'm going to then share. That's weird. Is it still sharing? It looks like it's still sharing.
Share a window instead of your screen.
And then, so this is the ROC curve from the cross-validation for this particular family. It is, not perfect. The nitrogen families that Christine were talking about were much better behaved. That's because this is a pretty difficult problem. As I talked about before, the structural similarity with the sequence variation is a tricky problem, and it's surprising actually that this can kind of capture that. Another thing that we're using is these long hydrophobic hydrophilic K-mers. And what we're thinking that is, as Christine was talking about, what we're thinking that actually captures kind of more of the general patterns that you're seeing rather than the details of the sequence at each point. And so the ROC curve, just to very briefly, if this ROC curve went all the way up to the corner and all the way over, you would have classified all of your positive examples perfectly, all of your negative examples perfectly. If it followed this dotted line, you would have been at random chance, again, like that K of 4 with the hydrophobic hydrophilic just does random chance. This does, fairly well. I was able to replicate the results that I'd gotten in the paper with a lot of hard work. In the paper, a lot of hard work with Snekmer in just a very quick amount of time. I mean, we already knew what the right parameters were to put in, so we're a little bit ahead of the game there. So I'm just going to run a search, and then maybe we can I'll show the search results and I'll show the cluster results, and then maybe we can show some of the documentation. Over that. And I'm going to go back to which terminal am I going to? Is that the right one? It is, okay. So I'm gonna do a search, and I have to do a couple of, oops. As I said, I'm gonna streamline this a little bit so it's more usable. So I'm going to So I've made these models, and now I'm going to use these models. And I have to put them in a separate directory, but that's something also we're working on. And then I'm going to swap out, because we're using the same directory, I'm just swapping out the input file. We don't want to apply the model to the same stuff we just trained it on, because probably know the answer we're going to get. And so what I'm going to do is put that input there, and now we've got a couple of FASTA files that are just kind of randomly chosen, and we're going to apply this search. So that's, again, taking that ubiquitin ligase model and applying it. So it has basically taken the k-mers, scored them, and has a model that it can apply to the FASTA files. And that's going to take, again, a second here, and then I'll show you actually, you know what, I can just skip ahead and show you the results through the magic of television. Because I did this earlier just to make sure that it worked, and it's not that exciting to watch Snekmer run. Rob thinks differently. He really likes to watch Snekmer run.
Well, especially log files, just keep going for hours and hours.
Okay.
So just to clarify, you generated a model based upon the control and ubiquitin ligase files that you made.
Yes.
And then are you applying it to separate sequences?
To separate sequences.
Okay.
Yeah. Sorry, that wasn't clear. Yeah, I had basically pulled two genome. It's about 10 or 12,000 sequences, and they're just kind of randomly chosen just to demonstrate. And the results come out in this format, which are basically a tab delimited format. Based on your input, it lists the sequence ID, which is, not terribly useful in this case because it's just a uniprot sequence ID. And then the important things are the score in family. It will show up as true. I'll show you in just a minute. And the probability. So the probability of this being in the family versus it not being in the family. And if you, do an Excel sort, you get all of the true ones at the top, and we get some hits. I kind of chose this such that I knew that we'd get hits because these aren't necessarily, widely, I was going to say ubiquitous, but that's different. Ubiquitins. But I did this so that we get some actual hits. None of them are particularly high scoring. So if you see something that, if I would have put in, for instance, members of the training set, you'd get 99, 98, and we've done that before. And so that would be, you could go in and take a look and see what these proteins are and look at their annotations and say, oh, this is annotated as a type 3 secreted effector, but it doesn't have any functional annotation. So we know that maybe it's a ubiquitin ligase or something else like that, right? So the search is still ongoing, but I'm going to show you in the interest of time, because I definitely don't want to let us run out of time.
Well,
So
you should also point out that, again, this is a particularly thorny protein family. Your results are almost guaranteed to look better than this.
yes, that is true. This is a very, right, because of the sequence divergence that we know to be in this protein family. The nitrogen cycling families, we've also done it on a couple of, a few different protein families that we've identified for the SFA that we're working on. And that works really well, and you can identify those things. It's pretty clear.
Do you do any experimental validation, for candidates?
Well, that's the idea, yeah. So all of this, so have we done any experimental validation? We actually have experimental results from the PERCON SFA that weren't meant to validate this, but do that's a little bit of a circular thing, but we're planning on going forward and validating some of them,
No, but
we had some
that's
lab
very cool.
results.
It's like a good, it's, for me, I see it's like a good genome mining tool for
Yeah.
many different things.
Yeah, and part of the impetus for this was that, there are other tools that you can do this. You can certainly do this with Hammer, things like this. It takes a little bit more preparation and more knowledge that you've got actual like multiple sequence alignments and things like that that are in good shape. This doesn't require multiple sequence alignments.
So you can work with genomes maybe that are more complex and not complete or like.
Yeah, exactly. And so one of the places where we're thinking, Bill has talked about this a lot, where if we have incomplete protein sequences, we're likely to be able to match protein functions, protein families using this approach, whereas something like an HMM wouldn't do that because you basically wouldn't get.
Yeah, especially like for plant genomes with chromosomal.
Oh yeah, the duplications.
Duplication events, but it's very hard.
Multiplications. So I could run the clustering. It's Snekmer cluster. Let's just pretend that I did that. Poof. So those two, the search and the model and search are kind of paired, right? You'd run the model, you get the models, and then you'd search. We're also building models, so we'll have like a database of populated models for different kinds of purposes. Right now, we've got a handful, but we're working on putting more together. The cluster, as Christine said, is more for when you don't know what that structure is. You want to look at, some different genomes, or you want to look at a sequence collection and you want to say, what's similar here? And then what do we do with it from there, right? We could use those clusters to build models, is what we've talked about. So this is output from clustering. It looks pretty similar. You have the sequence ID. You have the main important thing here is you've got a cluster number. It's very similar to other clustering methods where you have some output of some cluster, and it doesn't have a graphic, or I don't have a graphic output up here, but you can basically smallest to largest, whatever. And so you get this group of sequences, and I don't know off the top of my head what these are, but when I go through with other examples where I have the information, it's things that you would expect to be in clusters. And we've actually done this on a really large scale as well.
Is this the 10 genomes?
This is actually 4 genomes from that 10 genomes. Yeah, we used some of Mahantesh's clustering tools to do a 25 million sequence cluster that we're still kind of waiting to analyze, but we've got a lot of clusters and a lot of information. Yeah, so okay, so do you want to take a look, or do you want to look at the read the docs really quick?
Yeah, just go through a little bit of the code repo and if anyone has any questions or.
And we'll have you sit in front of the camera here, and I will share the appropriate screen.
So those cluster results were that was a different running of Snekmer that you had done with a different set of.
Yeah, sorry. Yeah, well, so that was basically the sequences that I searched, those random genomes that I pulled down. As an example, I basically just put those in to cluster them. That's a good point. It's separate from the ubiquitin ligase story. It's just showing how you can run Snekmer in the cluster mode. It's a pretty different kind of application, right? You have no a priori knowledge of what those families might be, and you want to say what in this group of sequences is similar to each other.
So do you need the same kind of you don't necessarily need a control file in the same way, and you only need FASTA
You only
formatted
need FASTA
proteins.
files, yeah. So you could dump any FASTA file in there and get clusters out. It's, similar to some other tools, MMSeeks2 being one of them, where you can do that. And as Christine showed, we did the comparison and found that we actually get kind of an orthogonal set of clusters that are equal in quality in terms of what they group together as annotations.
So a question can follow on. You mentioned that if you vary the K, you get different numbers of clusters, but they may also have significance both on the high or the low side, and that was why you left that up to the user. Could you talk a little bit about why that is, or maybe hypothesize this?
Yeah, I mean, we've explored that more with the model mode with for instance, the nitrogen cycling families example. But in those cases, I have sort of a high level heat map that looks at each individual family and how those models perform across different parameter sets. And interestingly, not all families perform best under certain conditions. I have that heat map that compresses all that family information. But certain families, certain alphabets and K combinations perform really well. And those same parameters perform poorly for other families. And so that we're thinking, our conclusion there is that the parameter set that you use to build your models can be very important and can, encode different types of information that may be more or less important for different protein families. And that's why we decided flexibility for the user is something that we want to sort of build into the tool so that the user can, for instance, if they have a specific protein family that maybe doesn't work as well under, say, a standard parameter set, they have the ability to explore other parameter sets. But obviously there are some parameter sets that just work terribly for all of them because the K-mers are just so common, and yeah, essentially you're just cutting up the sequences into non-informative, overly non-specific pieces. Yeah, you can think of it as image compression almost. So different image compression methods might preserve different aspects of an image. So if you navigate to the code base on GitHub,
It's the other way. It's okay, I have as much trouble going the opposite direction because my kids have it, and I'm like, why do you have your scroll?
Okay, it's the wrong way, but this is what you'll find. So we have that high-level summary image and sort of a brief description, a high-level description of each of the modes, which I've gone into in my talk as well. For more information, we've also built a documentation page to hopefully help the user, get Snekmer set up and working a lot easier. And so, I thought I clicked it. It's going.
Yeah, I'll have to see.
It's loading, I think.
Yeah, and the neural network is very suspect this afternoon.
Yeah, so if you click on the link to the official documentation, you'll find this here. We, sorry, have, scrolling the wrong way again. Information, high-level information, and sort of information on how to get Snekmer installed on your system, which will also take a very long time. Oh, actually not that bad. We have instructions for installation via Conda through just environment YAML files, so that'll just set up all the dependencies of Snekmer for you. And then you just need to activate your environment, you're ready to go. So, and then, yeah. We also have more information about, the config file that Jason shared. There's a lot of parameters in there, and that's because we wanted, again, to give users as much flexibility as possible. For instance, you can choose your clustering algorithm. We've encoded several types, and you can choose the parameters that you use to set up your clusters therein. But we've included, these descriptions. K and alphabet are probably self-explanatory, but also, for instance, if you have odd file extensions that aren't inherent, you can specify those. I think as default, we have FASTA, FAA, FNA, so. And then other parameters as well for the model and cluster, so you can just go here and refer to that. And then, just information on usage and configuration.
It's important to note, it does only work on protein sequence.
And then we also have descriptions of the alphabets as well, and you can refer to the linked citations, but these are the 6 standard encoded libraries. They're encoded 0 through 5. And if you choose to use use no encoding, you can also choose the none option, which is this one.
John's question was a really good one about user-defined alphabets, because it's a pretty simple thing. We've talked about adding the feature, we just haven't gotten around to it. So
yeah, actually, if you look if you explore around the Snekmer code, you'll actually see in the alphabet module, we have sort of a vestigial function for defining your own alphabet, but we haven't fleshed that out. But that's definitely something that we've talked about and we'd like to include at some point in the future. But for now, we have these 6 encodings, as well as 1 non-encoding. Yep. Any questions?
we had
2 minutes.
Yeah.
Okay. Yes, we are about on time, so that's about it. Yeah, Snekmer is sort of an ongoing work as well, and if you find yourself using Snekmer in any application, we love to hear feedback. We do also have we actively monitor the GitHub issues as well. So
we have many ourselves.
Yeah, we do track a lot of intended features or bugs. Everything is through GitHub, so feel free to leave a note on there as well if you run into your own issues. But for now, we have we have a working prototype that we're working on adding improvements to.
I'm really interested in the clustering, like use of Snekmer for clustering, for understanding like highly paralogous protein families where, like for serine hydrolases, you have a genome might have 25 serine hydrolases, and the question is, what's the substrate for each one? And so that's where if you get the clusters right, then each cluster might have one substrate and a different cluster have a different substrate.
Yeah, and you can automate. So we've got scripts they sit on top of Snekmer, but we've done this on the cluster where we've automated these sweeps through different parameter spaces. So if you have something like a set of serine hydrolases and you know something about what substrates they have, we could basically generate 50 different clusters based on all these different encodings and see which one's the best, right? Because you don't know a priori going in, you don't know which one might work the best.
Yeah, and you might know that this little group of hydrolases has this substrate, but the other 24, you don't know what the substrate is.
Yeah.
So you might have some idea of how tightly or not to cluster things. But that's why I think that the different alphabets might sort of reveal these concealed differences that you don't see just from the total sequence. Because you can do clustering. There's this thing called the sequence similarity index that John Geralt from University of Illinois or something has, that a lot of people use, that makes clusters, but it's just based on sequence alignments. So it might.
We should talk.
Yeah.
So we've also got I don't know how well it's implemented right now, but the concept of the background set, where when we started out, we were just throwing in just the protein sequences, and so the algorithm was unaware of any other usage of those k-mers beyond inside those families, and we were like, oh, what we really want is background set, so you can find out which k-mers are truly informative towards the family. And you might be able to do some clever things deals with, you know, I'm interested in this family, but not these ones, so use that as the background set, and this one's as my target set, and then that might be able to identify the K-MERS that are really unique to that. Could more precisely define the families or something.
Well, I have some examples that might be good, like test cases, or
Like test
see how it
cases.
works. Yeah.